Welcome. Today we're talking about tumor suppressor genes in Chapter 10, and I have this divided into two parts. In the first part here, we're going to talk about basic definition of tumor suppressor genes and discuss how you can end up with mutations in these genes that could potentially lead to cancer. In the second half, I'm going to focus primarily on examples, prominent examples of tumor suppressor genes, and then finish it off with a little summary at the end. All right, so I know we've talked about this before, but tumor suppressor genes are really sort of the yin to oncogenes yang, so to speak, where oncogenes are those that help drive cell division, and if we gain function in tumor suppressor genes, that can lead to cancer. Oncogenes. And in tumor suppressor genes, these are going to be genes whose loss or inactivation can lead to cancer. And the normal role of tumor suppressor genes is to inhibit cell division or to promote cell death, or I'll add a third here, or to basically help maintain the health of your DNA. And so the idea that tumor suppressor genes even existed, or even a thing, came from some cell fusion experiments. And what researchers were doing was in the lab fusing cancer cells with normal cells. So you end up with this weird fusion cell that temporarily has two separate nuclei. And at some point, those nuclei will fuse together, and you have this true hybrid cell that will initially have normal growth. So it's as if the normal genes from the normal cell nucleus are overriding the cancer cell genes. But because this is essentially a tetraploid cell, or it's going to have four copies of all the chromosomes in this hybrid nuclei, the cell doesn't really know what to do with that. It doesn't like that, so it will start shedding, spitting out chromosomes, basically. So you get this chromosome loss. And what researchers noticed was as this phenomenon of chromosome loss began, we would see some relatively high percentage of these hybrid cells now switching over into uncontrolled cell growth, basically into that cancerous state. So it was as if those hybrid cells were losing some DNA that was helping to keep the brakes on cancer, right? So they named those tumor suppressor genes. And of course, researchers and clinicians were very interested in, well, what are those genes? And so researchers just started searching for them, basically kind of by process of elimination. So the first that were identified were really those associated with some of those inherited cancer syndromes that we talked about in Chapter 8. You may remember a lot of those genes that we saw in Chapter 8 were tumor suppressor genes. And initially, they just looked for sort of larger chromosomal abnormalities, so, deletions in a chromosome or something that might be associated with one of these inherited cancers. The idea being, okay, what region of a chromosome is missing if this is a tumor suppressor gene, and we know this particular region is missing, what genes are actually in that missing region, and start to narrow it down from there until we can get locked into a specific gene and say, well, this has got to be it. This is the tumor suppressor gene, and sort of figure it out with that kind of detective work. And as I mentioned, to have a cancer being caused by mutations in tumor suppressor genes, they really need to be loss of function mutations, right? We need to completely cut the brakes and eliminate the function of any protein coded for by the tumor suppressor genes. So in most instances, that means the cell needs to be homozygous for mutations in a tumor suppressor gene, because we have diploid cells, right? Two copies of every tumor suppressor gene, two copies of every chromosome. We would need to mutate both copies typically for it to become a problem. And so geneticists will refer to this process of becoming homozygous as loss of heterozygosity, right? Usually being heterozygous for a mutation is not usually a problem, but once you become homozygous, you lose that heterozygosity. It can be an issue. And so there's a few ways that could happen. Just to orient you here, here is a cell that is heterozygous for a mutation in tumor suppressor genes. So we've got our homologous chromosomes, each chromosome made of two sister chromatids here because we're getting ready to go through mitosis. So one copy of the tumor suppressor gene is mutated, denoted by the little red flares here. The other copy is normal. And because we have that one normal copy in every cell that's produced, these cells are normally functioning. Um, so one way we could become homozygous or lose the heterozygosity and create a problem is through mitotic non-disjunction. So you may remember non-disjunction is when the chromosomes or the sister chromatids do not separate properly. And so in this instance, both normal copies, both normal sister chromatids end up going to the same daughter cell. So the cell on the left here has no normal copy. So even though technically it's, I guess considered hemizygous, we've lost that heterozygosity. We don't have the normal copy of the tumor suppressor gene that we need. Another possibility would be mitotic recombination. Usually we think about recombination as a phenomenon that only happens during meiosis, right, when the homologous chromosomes pair up, but it can and does happen during mitosis as well. So if you have a recombination event between homologous chromosomes that happens to involve that mutated tumor suppressor gene, now we end up with a situation like this, where if we follow it through to the end of cell division, now both of the mutated copies have ended up in the same daughter cell. So we've lost both copies of the tumor suppressor gene here. And then the third possibility is what's called gene conversion, and I'll show you on the next slide a little bit more detail. But this comes down to a phenomenon that can happen during the copying of the DNA, basically before we even get into mitosis. So during S phase, as we're going through DNA replication, and it looks something like this. So these would be our two homologous chromosomes here, and imagine these are tumor suppressor genes, and the green copy has a mutation, the red copy is normal, so we're heterozygous for that mutation in this cell. And DNA polymerase is copying the normal version of the tumor suppressor gene here. And in gene conversion, what happens is sometimes the DNA polymerase will jump from one homolog to the other and copy some of that information over as it's carrying out DNA replication. So if it happens to jump over to the homolog that has the mutated tumor suppressor gene and copy that mutation over into the normal version, then we go back. Now we're ending up with something like this, right, where we basically have 3 copies of that mutation. And so it's very likely we're going to end up with at least one of the daughter cells where we've got 2 mutated tumor suppressor genes. So this is just speaking to mechanism, right? These are ways that we could knock out both copies of the tumor suppressor gene. What's actually causing the mutation, with the exception of gene conversion, going to be things we've already discussed. So, chemicals, radiation, infectious agents in some cases. Another phenomenon that could reduce the expression or potentially even eliminate the expression of tumor suppressor genes without actually mutating the genes themselves would be some of these epigenetic phenomena, things like methylation or changes in acetylation pattern. But we're going to focus primarily on DNA methylation, which, as it sounds, is literally just adding or removing methyl groups, chemical methyl groups, from the DNA. And in particular, there are enzymes called methyltransferases that like to add methyl groups to cytosine nucleotides that are adjacent to guanine. So anytime you have cytosine C next to G in the DNA, that's a prime site for potential DNA methylation. And you combine this with changes in histone acetylation, this can really change the packaging of the DNA and its ability to be able to be transcribed and hence translated. And so this process of DNA methylation is carried out by enzymes called methylases. That makes sense, right? And there's really two categories of methylases. There are what are called de novo basically meaning brand new. De novo methylases are going to add methyl groups to previously unmethylated cytosines. Um, and then we have what are called maintenance methylases, which are sort of maintaining existing methylation through rounds of DNA replication. So imagine you've got your DNA here where you have these cytosines on both strands that are methylated. We separate the two strands for DNA replication, synthesize our new DNA, and the new DNA by default is not going to be methylated. But then along come the maintenance methylase enzymes, and they realize, oh, yep, that should have been methylated, that should have been methylated. So we're just maintaining that existing methylation pattern. And I already mentioned this word, but this would be considered an epigenetic change. So we're not actually changing the DNA sequence anywhere in any way, but we are altering DNA expression patterns with these methylation events, as I'll get into. And these methylation patterns can actually be inherited, at least as we're seeing here, from mother cell to daughter cell, and in some cases potentially even through the gametes from one generation to the next. And so researchers have been interested in this phenomenon in cancer cells, and what they see is that overall, like globally, methylation actually decreases in cancer cells. There's less methyl groups on the DNA overall, but we actually get increased methylation in the promoters of specific genes. And so what you're seeing here in this example, here's a normal cell. Let's say this is a tumor suppressor gene and its promoter, and these little black tags are representing CPGs that could potentially be methylated, but the black are not methylated. So we're seeing sort of low numbers of methyl groups in the normal cell. But when we look in the cancer cell, we're seeing just a ton of methylation in the promoter for this gene, and that basically has the effect of shutting down transcription. So RNA polymerase shows up to try to transcribe the gene, and it doesn't like the look of all these methyl groups on the promoter, and transcription is essentially turned off. So we've basically lost the function of this tumor suppressor gene, even though there's the gene sequence itself is fine. There may be no mutations there, but we're not making the protein, so we could consider it a loss of function. Just to show you some data from one particular study, researchers here were looking at methylation patterns in one particular RAS gene, in the promoter of this gene specifically. And what they found was if they look at the RAS promoter in a cancerous tumor, pretty much every cytosine that can be methylated is methylated. So here every circle represents a potential methylation site. Blue means it's methylated, white means it's not, right? So very high levels of methylation in the RAS gene promoter inside the tumor. If we look just next to the tumor in what still looks to be normal tissue, we see a slightly increased amount of methylation in that promoter. And if we go even farther out into completely normal tissue, or looking at tissue from actually another individual here that's not cancerous, we're seeing very, very low or non-existent methylation. So we're seeing that correlation between the pathology and the intensity of the methylation in these specific promoters. And because there's been a lot of interest in this, people have looked at methylation patterns in lots of different cancer types and for lots of different tumor suppressor genes. And so what you're seeing here are sort of these signatures that exist for different types of cancer. So in this graph, you have different types of tumors on one axis, and then some common tumor suppressor genes on the other axis. And you can see, if you look cancer by cancer, we're sort of seeing different signatures in terms of which tumor suppressor genes tend to be hypermethylated, and this is likely specifically the promoters for these tumor suppressor genes. So we know this is tied in with helping drive some of these specific types of cancers. And one reason there's been a lot of interest in this is methyl groups can be added or removed with simple enzyme treatments, right? So theoretically, we might be able to go in and demethylate promoters to help treat some of these cancers or slow cancer growth down. So there may be some clinical application to our understanding of this whole phenomenon. Typically, researchers will look at methylation patterns by doing sort of a modified DNA sequencing technique that will allow them to see which nucleotides are methylated or not. But there are actually stains that would allow pathologists in a lab, for example, to see methylation patterns. And that's what you're looking at here. So this purple stain, basically the more purple you see or the more darker color you see in these images, the more methylation there is. And we've got cervical cells on the top, breast cells on the bottom. The images on the left are either low-grade tumor or completely benign samples. And then on the right, we have cancerous samples. And you can see that increased methylation is very obvious just from this simple staining procedure. So this could be another toolkit in terms of the cellular pathology or even diagnosing certain cancers, grading and staging. Could be useful there. All right, I'm going to stop there and we will pick this up in the second half.